I became interested in sea lions as ecosystem samplers when I was introduced to a quarterly time series of California sea lion diet that NOAA's Southwest Fisheries Science Center has collected from rookeries in the Southern California current since 1981. This diet survey holds a wealth of information about how the forage base has varied seasonally, spatially, and on multi-annual and decadal scales. Studying the diets of large marine predators provides essential information for ecosystem modeling and ecosystem-based management. Just as importantly, large marine predators integrate prey over large areas, so they also can serve as valuable ecosystem indicators, particularly of forage community composition and individual prey species abundance. Unlike most other large marine predators, many pinniped species make frequent visits to haulouts or rookeries to rest or nurse their young, effectively bringing back information from their foraging trips to deposit on land in the form of scats. The accessibility and non-invasiveness of pinniped scats allows for temporally and spatially intensive studies, such as Southwest Center's time series, that are able to resolve ecosystem variability and elucidate links between trophic and population ecology. However, to unlock the full potential of these data, we need to account for differential recovery of prey hard parts in scats, which can vary tenfold or more among different taxa and prey sizes, particularly for otoliths from teleost prey. Captive feeding studies can provide correction factors for specific prey species and sizes, but the taxonomic and size diversity of California sea lion prey render this method intractable as a standalone approach. Previous work, based on captive feeding studies, showed that morphometrics of fish otoliths, such as their length, width, and mass, could explain some of the variation in their recovery rates. Seizing on this potential to predict recovery rates for species and sizes not included in captive feeding studies, one of my co-authors developed an extensive prey hard parts database of otolith morphometrics, covering most of the teleost prey taxa of California sea lions across a range of sizes. In this analysis, we combined two data streams. One, data on recovery rates of known species and sizes of some common prey taxa from two captive feeding studies of California sea lions, and two, corresponding predicted morphometrics for the otoliths in the captive feeding studies based on regressions from the prey hard parts database. We developed a binomial mixed effects model that could predict recovery rates of novel prey species and sizes from morphometric indices. We used a leave one prey species out cross-validation approach to find the model that best predicted novel species recovery rates across taxa. We also separately reassessed recovery rates of squid beaks, which are resistant to digestion but are sometimes regurgitated rather than defecated, using an intercept-only binomial mixed effects model. Based on these models, we can now reconstruct relative consumption rates over a much broader set of prey taxa and sizes than could be achieved from captive feeding studies alone, allowing better reconstruction of the taxonomically diverse diet of California sea lions. The richer resulting time series of sea lion diet can support more robust analyses of prey and size selectivity, in turn better informing ecosystem models, and also allows stronger inference about forage community and individual prey species dynamics from the sea lion diet time series outside of seasons when traditional fisheries surveys are conducted. The captive feeding studies on which our analysis is based omitted some important parameter space for otolith morphometrics, including that for young-of-year forage fish, such as anchovy and sardine. Additional captive feeding studies to expand this parameter space would further improve accuracy and robustness of estimated relative biomass consumption across age classes of important forage species. The recovery rate models resulting from this analysis are currently being used to reconstruct time series of relative biomass consumption across prey taxa and sizes by sea lions, for use in a multitude of other applications, such as characterizing the link between trophic and population ecology for predators and prey, and developing ecosystem models that forecast population outcomes under a range of climate change scenarios.